So you'd like to know how to extract chloroplasts from plant tissue? Well, I'm the guy to ask, and really, it all boils down to two things: cell fractionisation and ultracentrifugation. And I know, two very exciting words. They could really knock your stalks off. Unbelievable. I'm Professor Ulrich von Lichtenstein of Gelderland, and welcome to today's lesson. Step 1 is breaking the cells open. Emotional damage can get the job done, but I think physical damage, like using a blender, is best. And this term, breaking open the cells, is called homogenization, and this is a leaf. This goes into the blender along with a solution, a very special solution. A solution which is cold, isotonic, and buffered. But what do these terms mean, I hear you ask? Well, let's go over them. It is cold to reduce enzyme activity, because when the cells are broken open, or homogenized, if we want to use our key term, then enzymes are released which can damage the organelles. It is also isotonic, which means there's an equal water potential, so water does not leave or enter the organelles and damage them. It is also buffered to maintain a constant pH to not damage the organelles. And as you may have heard, but you really need to pick up on, is that I said organelles and not cells. Then enzymes are released which can damage the organelles, so water does not leave or enter the organelles and damage them. It's easy when going over these factors in an exam to say cells instead of organelles, but we've already broken open the cells, homogenized them, and these factors are to do with the organelles. So now that we have our cold, isotonic, and buffered solution, I knew you were all going to say it, it is now filtered to remove large debris and it is ready for part 2. Ultracentrifugation is how we separate out the different organelle types. But how is this done, I hear you ask? Well, here is an artist's interpretation. That's right, the centrifuge spins and separates out the organelles by applying centrifugal forces, which means that the denser organelles form a pellet at the bottom, as shown in this diagram. The liquid with the remaining organelles is called the supernatant, and this is poured off and spun again at higher speeds. Here is an artist's interpretation. And this process is repeated until all the organelles are separated. But for what you're looking for, it only takes 2 spins of the centrifuge to separate the chloroplasts. Because, and I don't want to be mean here, chloroplasts are pretty dense. You are now free to remove the pellet and examine it under a microscope, or just sit back and celebrate a job well done. Because you now understand how to separate chloroplasts from plant tissue. Congratulations.